Good morning, students. I, myself, Dr. V.B. Kadam, Associate Professor, Department of Botany, KTHM College, Nasik. Students, today we are going to learn the chapter Photosynthesis, and the topic is Pigment System first and Pigment System second, its composition and light harvesting mechanism. That means the initial phase of photosynthesis. It is also known as light dependent part of photosynthesis. Now first we are going to learn the Emerson's red dwarf effect and enhancement effect. The Emerson's effect is the increase in the rate of photosynthesis after chloroplasts are exposed to light of wavelength 670 nanometer. Now 670 nanometer of light content a red color and 700 nanometer it content far red color of light. That means alternate fashion of light for few hours 670 nanometer of light given to the chloroplast, and then for few hours 700 nanometer of light. That means alternate fashion of red light and far red light. When simultaneously exposed to light of both wavelengths, the rate of photosynthesis is far higher than the sum of the red light and far red light photosynthesis rates. Wavelengths beyond 700 nanometer are apparently of insufficient energy to drive any part of photosynthesis. So a huge drop in efficiency has been noticed at 700 nanometer of light. This phenomenon is called as a red dwarf effects. Now in 700 nanometer of light having the red color in visible spectrum, and now the 700 nanometer of light content red color. That's why this effect is known as a red dwarf effect. That means the 700 nanometer of light given to the chloroplast, the yield of photosynthesis is suddenly decreased, and that's why this effect is known as a red dwarf effect. In other words, there is a sharp decrease in quantum yield at wavelength greater than 680 nanometer. This decrease in quantum yield takes place in the red part of the spectrum. The number of oxygen molecules released per light quanta absorbed is called as quantum yield of photosynthesis. That means how many amount of oxygen evolved and how many amount of light rays captured by the plants. This effect was first of all noticed by Robert Emerson in 1958 on chlorella. Chlorella is algae. Studying the rate of photosynthesis at different wavelength of light, that means from 390 to 760 nanometer of light. Now this range of light is known as visible light. It is useful for photosynthesis. Monochromatic light of different wavelength is used, and rate of photosynthesis is measured. The sudden fall in photosynthetic yield in the far red region, that means greater than 680 nanometer of light, compared to the red region of the electromagnetic spectrum, is called Emerson's red dwarf effect. The increase in photosynthetic yield by the combined effect of rate, that means 680 nanometer of light, and far red light, that means 700 nanometer of light, is called Emerson's enhancement effect or Emerson's effect or improvement of the yield of photosynthesis. Friends, look at these pictures. This is the Emerson's red dwarf and enhancement effect pictures. Now this is the red light, the wavelength is 680 nanometer of light. The 700 is far red and 680 plus 700, that means alternate fashion of light, red plus far red. The yield of photosynthesis is suddenly increased. Now the rate of photosynthesis is in red light, in far red light, but both giving alternate fashion, red, far red, it gives higher yield of photosynthesis. These works provide experimental evidence for the presence of two photosystems, that means photosystem first and photosystem second. See, the picture is electromagnetic spectrum. Now, sun emits the various radiations. It contains radio waves, microwaves, infrared waves, ultraviolet, X-rays, gamma rays. Now, very few amount of rays is known as visible spectrum of light, and visible spectrum of light is present in between infrared and ultraviolet rays. Now this visible light having the range between 400 nanometer to 700 nanometer of light, it contains 7 various colors, different colors. And now these 400 nanometer to 700 nanometer of light is useful for photosynthesis. That's why this light is known as visible light of spectrum. Pigment systems, it is also known as photosystems. The discovery of red dwarf and Emerson's enhancement effect has led scientists to suggest that photosynthesis is driven by two photochemical processes: the absorption of light energy and its conversion into chemical energy occurs in multi-protein complexes called photosystems located in the thylakoid membrane. These processes are associated with two groups of photosynthetic pigments called as pigment system first and pigment system second. Wavelength of light shorter than 680 nanometer affect both pigment systems, while wavelength longer than 680 nanometer affects only pigment system first. Pigment system first is relatively very weakly fluorescence, while pigment system second strongly fluorescent. In green plants, pigment system first contains chlorophyll B, different forms of chlorophyll A such as chlorophyll A 670, chlorophyll A 680, chlorophyll A 695 or chlorophyll 677, chlorophyll A 684, chlorophyll A 692 or chlorophyll A673, chlorophyll A683. These differs according to different workers. A very small amount of a special form of chlorophyll A absorbing at 700 nanometer of light, which is known as P700, however, constitutes the reaction center of pigment system first. The pigment system second contains chlorophyll B and some forms of chlorophyll A, such as chlorophyll A 662, chlorophyll A 670, chlorophyll A 677 nanometer of light. A very small amount of special form of chlorophyll A called P680 constitutes the reaction center of pigment system second. In some plants, P690 constitutes the reaction center of pigment system second. Carotenoids are present in both the pigment systems. Each pigment systems or photosystem constitutes of a central core complex and a light harvesting complex. Now the role of light harvesting complex, it is also known as antenna complex. They absorb the specific wavelength of light and absorb light transferred towards the core complex. In pigment system first and pigment system second, these are called as core complex first, light harvesting system first, and core complex second, and light harvesting complex second, respectively. The light harvesting complexes contain antenna pigments associated with proteins. Their main function is to harvest light energy, that means to absorb the solar energy and transfer it to their respective reaction centers. The core complexes consist of their respective reaction centers associated with proteins and also electron donors and electron acceptors. The two pigment systems, pigment system first and pigment system second, are interconnected by a protein complex called cytochrome B6F complex. The other intermediate components of electron transport chain are plastoquinone and plastocyanin, acts as mobile electron carrier or electron transporter between the complex and either of two pigment systems. In case of the red and blue-green algae, the pigment systems contain phycobilins in place of chlorophyll B. The relative proportion of chlorophyll B and different forms of chlorophyll A is different in the two pigment systems. Plastoquinone, which in fact is a hydrogen carrier, acts between pigment system second and cytochrome B6F complex. Plastocyanin acts between cytochrome B6F complex and pigment system first. The light energy absorbed by other pigments is ultimately trapped by P700, that means reaction center first, and P680, that means the reaction center 2, forms of chlorophyll A, which alone take part in further photochemical reaction. Pigment system first complex consists of 200 chlorophylls 50 carotenoids, and a molecule of chlorophyll A absorbing light at 700 nanometer, and this constitutes the reaction center of photosystem first. The pigment system second complex consists of 200 number of chlorophylls, 50 carotenoids, and molecules of chlorophyll A absorbing light at 680 nanometer called P680 at the center. This constitutes the reaction center of pigment system second. Chlorophyll A is present in both component of a photosystem. That means photosystem first and photosystem second. The absorption of light energy and its conversion into chemical energy occurs in multi-protein complexes called photosystems located in the thylakoid membrane. A photosystem has two closely linked components: an antenna containing light absorbing pigments and a reaction center comprising a complex of proteins and two chlorophyll A molecules. Each antenna complex contains one or more light harvesting complexes. The energy of light captured by light harvesting complexes is funneled to the two chlorophylls in the reaction center, where the primary events of photosynthesis occurs. Found in all photosynthetic organisms, both eukaryotics and prokaryotics, chlorophyll A is the principal pigment involved in photosynthesis, being present in both antennas and reaction centers. In addition to chlorophyll A, antennas contain other light absorbing pigments: chlorophyll B in vascular plants and carotenoids in both plants and photosynthetic bacteria. The presence of various antenna pigments which absorb light at different wavelengths greatly extend the range of light that can be absorbed and used for photosynthesis. Now, this is the structure of beta carotene. Photosynthesis at different wavelength. Students, look at the pictures. Here is the absorption spectrum, and on x-axis is the wavelength of light. Now, the chlorophyll B absorbs the light in 500 nanometer of light. Chlorophyll A absorbs the light in between 400 and 500 nanometer of light, and also in between 650 to 700 nanometer of light. One of the strongest pieces of evidences for the involvement of chlorophyll and beta carotene in photosynthesis is that the absorption spectrum of this pigment is similar to the action spectrum of photosynthesis. See, this is the action spectrum and this is the absorption spectrum. The absorption spectrum of beta carotene and chlorophyll A is the same in action spectrum. The latter is a measure of the relative ability of light of different wavelength to support photosynthesis. The action spectrum of photosynthesis in plants, that is, the ability of light of different wavelength to support photosynthesis. The absorption spectrum of three photosynthetic pigments, chlorophyll A, chlorophyll B, and beta carotene. Each spectrum shows how well light of different wavelength is absorbed by one of the pigments. A comparison of action spectrum with the individual absorption spectra suggests that photosynthesis at 680 nanometer is primarily due to light absorbed in the antenna complex by chlorophyll A at 650 nanometer to light absorbed by chlorophyll B and at shorter wavelength to light absorbed by chlorophyll B and by carotenoid pigments including beta carotene. When chlorophyll A absorbs visible light, the absorbed light energy raises the chlorophyll A to a higher energy state termed an excited state. Now what happens, students? Antenna complex absorb the specific wavelength of light, and now this absorbed light is handed over or transferred towards the core complex. Core complex is present in chloroplast, and now the core complex is controlling 50 to 60 number of chlorophyll molecules. Now the harvested energy captured by antenna complex then transferred towards the core complex. Core complex decide one chlorophyll molecules to excite or to take this radiant energy. Now this condition is known as the excited state of electrons. This differs from the ground state largely in the distribution of electrons around the carbon and nitrogen atom of the porphyrin rings. Excited states are unstable and will return to the ground state by one of several competing processes. For chlorophyll A molecules dissolved in organic solvents such as ethanol, the principal reaction that dissipate the excited state energy are the emission of light, fluorescence and photofluorescence, and thermal emission, that means heat. The situation is quite different when the same chlorophyll A is bound to the unique protein environment of the reaction center. See, this is the structure of chlorophyll A. Chlorophyll B differs from chlorophyll A by having CHO group in place of the CH3. See, this is the structure of chlorophyll A. Now in place of CH3, CHO is present in chlorophyll B. In the porphyrin ring, a highly conjugated system, electrons are delocalized among three of the four central rings, and the atoms that interconnect them in the molecules. In chlorophyll A, magnesium ions rather than an ferric ions is in the center of the porphyrin ring and an additional 5-membered ring blue. In place of magnesium, ferric ion is present in chlorophyll B. Is present. Otherwise, its structure is similar to that of heme found in molecules such as hemoglobin and cytochromes. The hydrocarbon phytol facilitates the binding of chlorophyll to hydrophobic region of chlorophyll binding proteins. Absorption of quantum of light. The absorption of quantum of light of wavelength 680 nanometer causes a chlorophyll A molecule to enter the first excited state. The energy of such photons increases the energy of chlorophyll A by 42 kilocalories per mole in the reaction center. This excited state energy is used to promote a charge separation across the thylakoid membrane. An electron is transported from a chlorophyll molecule to a primary electron acceptor, the plastoquinone or quinone, on the stromal surface of the membrane, leaving a positive charge on the chlorophyll close to the lumen surface. The reduced primary electron acceptor becomes a powerful reducing agent with a strong tendency to transfer the electron to another molecule. The positively charged chlorophyll A, strong organizing agent, will attract an electron from an electron donor on the luminal surface. These potent biological reductants and oxidants provide all the energy needed to drive all subsequent reactions of photosynthesis. Electron transport, ATP synthesis, and CO2 fixation. Look at this picture. The primary event in photosynthesis, that means light harvesting process. Now this is the stroma portion, this is the thylakoid, and this is the lumen. Now what happens? The various wavelengths of light absorbed by this antenna complex. It is present in chlorophyll. It is also present core complex. After harvested light is transferred towards the core complex, and core complex decide each and every chlorophyll molecule's outer orbit having two electrons, and both electrons are spin opposite directions. Now this harvested light captured by this antenna complex and transferred towards the one molecules and its one electrons. Now these electrons taking this solar energy, radiant energy, and moves to transfer this energy. Now that process is known as excitation of electron molecules. Now these electrons is accepted by primary electron acceptor quinone, and now this quinone accepted is strong reducing agent electron donor. Now these accepted electrons transfer toward the electron transport chain from plastoquinone to cytochrome B6, then plastocyanin, then pigment system segment. This is the picture of photosystem, how photosynthesis photosystems are worked. Now this is the structure of thylakoid membrane. Now in thylakoid membrane, number of chlorophyll molecules are there. A reaction center. It contains core complex, antenna complex. Now this harvested light by the antenna complex transferred towards the core complex, and now core complex decide the electrons to take this radiant energy, and now these electrons are transferred from ground level to excitation level. That means taking radiant energy and it moves from the excited state. Home assignment for students: explain Emersons rate drop and enhancement effect. Number 2, describe pigment system first And second, and number three, what is electromagnetic spectrum?